Welcome. Take a moment to settle in, without trying to find the perfect position. Turn the volume down to the point where my voice is only just audible, a little below what would be comfortable in daylight. Nothing that follows needs to be remembered or understood or followed to the end. You can fall asleep at any point, and you will not miss a thing. Tonight we are going to talk about coral reefs. 100 facts to fall asleep. To know adverts, slowly, without hurrying. The sea withdraws slowly on a reef flat, and the withdrawal is more sideways than downward, so that what happens is less like water draining from a basin, and more like a sheet being drawn back from a wide bed, uncovering first the highest rubble near the shore, then the pale sand, then the ridged and pitted limestone that lies beyond, until a surface that was ocean an hour ago has become something a person can walk across in old shoes. On coasts with 2 high waters and 2 low waters each day. This happens twice, and around the new moon and the full moon. When the pull of the sun and moon act along the same line, the low waters go lower than usual and expose ground that stays covered for most of the month. Those are the mornings when people who live near reefs go out to look. The walk begins on loose material because the shoreward edge of a reef flat is generally a heap of what the reef has produced and the waves have broken. There are branches of coral snapped into finger lengths, tumbled until their sharp edges have gone, and among them the flat white plates of dead coralline algae, and shell fragments, and here and there a whole shell, and small round grains that are the worn-down remains of all of it. Underfoot, the pieces shift and clink with a light, hollow sound, dry and slightly musical, and the sound changes as the walk goes on, and the fragments become smaller and wetter and begin to pack down into something firmer. Then comes sand, and the sand is limestone too. Almost nothing on a coral coast is quartz. The grains are the skeletons of corals ground fine, the chalky internal plates of green algae that grow in little segmented stalks and fall apart when they die. The shells of single-celled organisms that build tests of calcium carbonate, and the spines of urchins, and the fragments of shells too small to identify. Held up to the light in a wet palm, the sand shows this mixture plainly. White and cream and faint pink, with occasional darker grains, and it feels different from beach sand made of rock. Softer in some places and sharper in others, and it dries very quickly and very white. Beyond the sand, the reef flat proper begins, and its dominant color at low tide is not blue or bright coral pink, but a sort of olive brown, because the exposed pavement is covered with turf algae only a few millimeters tall, along with mats and crusts of other algae, and all of it holds water like a sponge and squeaks faintly when trodden on. The pavement itself is old reef, cemented and leveled, and it is full of holes. Some are the wide, shallow depressions left where a coral head died and dissolved. Some are neat round tunnels bored by bivalves that spent their lives grinding into the rock. Some are the irregular passages of worms. Every one of them holds water, and every one of them holds something living, and the shallow ones dry to a crust of salt in the sun, while the deeper ones keep their inhabitants cool. The larger pools are the reason people cross the flat. A pool the size of a bathtub, left behind by the falling tide and floored with sand and rubble, becomes for a few hours a closed world that can be looked into from above without any equipment at all. Its water warms noticeably, several degrees above the open sea, and the animals inside it are the ones that can tolerate that warming. Small gobies rest on the sand with their pectoral fins propping them up, and dart under a stone at the shadow of a hand. Blennies sit in old wormholes with only their heads out, and their eyes swivel independently. Hermit crabs occupy the shells of dead snails and carry them over the sand with a sound like a pebble being dragged. Sometimes there is a small shark, patterned in dark blotches and no longer than a forearm, which is able to endure the low oxygen of a warm pool and can push itself over the rock on its paired fins from one pool to the next. Turning over a loose piece of rubble in a pool reveals more than looking at the surface ever does, because the underside of every stone on a reef flat is furnished. There will be encrusting sponges in orange and grey, flat and slightly rubbery, and colonial tunicates that look like transparent jelly with small dots in it, and coiled tubes of calcareous worms, and brittle stars with thin arms folded into the crevices, which unfold and pour themselves away with surprising speed when the light reaches them. There will be small crabs with heavy, blunt claws, dark-fingered, holding still. There may be a flatworm the width of a fingernail, gliding without any visible mechanism, and there will almost certainly be several kinds of snail waiting out the dry hours in the shade. The convention among people who do this often is to put the stone back the way it was, the same face down, because everything living on it is adapted to that orientation and will not survive being left in the sun. The corals still alive on the flat are the ones that can cope with heat, sediment, and brief exposure to air, and they have a characteristic look. Massive mounds of one common genus grow as boulders of dense limestone with a fine pitted surface, and where they have grown upward as far as the low tide will allow, the living tissue dies on the top and continues only around the sides, so the coral spreads outward into a low circular table with a dead flat center and a raised living rim. Such formations record the water level over decades, because the height of the living rim is set by the level below which the coral is never left dry. Elsewhere, there are small brown clumps of branching coral in the deeper channels, and encrusting corals lying tight against the rock, and soft corals that at low tide collapse into slack, drab lumps and give no hint of the shapes they take when covered by water. Among the sand patches lie sea cucumbers, which on many reef flats are the most obvious large animals present, sprawled in the open like discarded objects, some black and warty, some sandy pink and smooth. They pass sediment through their bodies continuously, taking out the organic matter, and the small coiled casts of clean sand behind them mark their routes. Handled roughly, certain kinds release sticky white threads that swell and cling and are very difficult to remove from skin, so they are best left where they lie. Giant clams sit wedged into the pavement with their shells gaping and their mantle tissue showing in patterns of brown and blue and green, and the mantle withdraws with a small jet of water when a shadow passes. Sea urchins crowd into crevices, sometimes several to a hole, their spines interlocking, and their grazing over generations has worn the surrounding rock into shallow basins. There are things not to touch, and they are worth knowing in advance rather than discovering. A stonefish resting in a pool is almost indistinguishable from the rubble around it, and carries venomous spines along its back. Cone snails, handsome and patterned, can deliver a sting from a modified tooth that extends beyond the shell. Fire corals, which are not true corals, raise smooth mustard-colored blades and crusts, and produce a burning welt on contact. Small octopuses shelter under stones and are best admired without handling. The general practice is to look with the eyes and use a stick or the flat of the hand, and to walk on bare rock and sand rather than on living coral. Further out, the flat begins to sound different. The steady hiss and thump of surf that has been in the background all along becomes closer, and the surface underfoot rises very slightly into a ridge of hard pink and purple limestone laid down by coralline algae, which are among the few organisms that thrive in constant breaking water. Beyond it, the reef edge drops away, and water pours back across the shallows in surges, each one a little further than the last. The pools begin to connect. The temperature drops. The animals that spent the low water, folded, closed, and hidden, begin to open, and the walk turns back toward the shore while the pavement is still visible under a hand's depth of moving sea. The pavement underfoot, the pink ridge at its outer edge, the branching thickets in the deeper pools, all of it is the work of an animal that is, taken one at a time, about the size of a grain of rice or a small pea, and in some species smaller than the head of a pin. A coral polyp is a soft cylinder of tissue, open at the top, closed at the bottom, and ringed at its upper rim by a circle of tentacles. It has no head, no eyes, no brain, no blood, and no skeleton on the inside. What it has, instead, is a single body cavity that serves as stomach and circulatory system together. A single opening that serves as both mouth and vent, and a sheet of cells at its base that lays down limestone beneath it for as long as the animal lives. Within the group that includes the reef-building corals, the tentacles come in multiples of 6, and a small polyp may carry 6 or 12, while a larger one carries 24 or 48, arranged in tidy rings. The soft corals and sea fans, which belong to a separate branch of the same wider family, carry 8 tentacles, each of them fringed along its sides with small side branches like the teeth of a comb, so that a magnifying glass is enough to tell one lineage from the other. In both cases, the tentacles surround a flat disc of tissue with the mouth at its center, often a slit rather than a circle, capable of gaping open wide enough to swallow prey a good fraction of the polyp's own size. The tentacles are not for grasping in the way a hand grasps. They are covered in stinging cells, tens of thousands of them in a single tentacle, each one a specialized cell holding a capsule under enormous internal pressure, and inside the capsule a hollow thread coiled like a fishing line and often armed with spines along its length. A trigger on the outside of the cell responds to touch and to chemical traces in the water, and when it fires, the capsule everts its thread, turning it inside out as it goes, and drives it into whatever has touched it. The discharge takes less than 1,000th of a second and is among the fastest movements produced by any living cell. Venom passes down the thread, the prey stops struggling, and the tentacles bend inward toward the mouth in a slow, deliberate fold that is easy to watch under a lens and almost impossible to see with the naked eye. Inside, the body cavity is divided by thin vertical partitions of tissue that run from the wall toward the center, increasing the surface available for digestion and holding. In season, the reproductive tissue. Prey is broken down first by enzymes released into the cavity and then absorbed by the cells lining it, and whatever cannot be digested is expelled back through the mouth. Small drifting food that never reaches the tentacles at all is caught in mucus on the surface of the polyp and moved along by the beating of microscopic hairs, which is why a coral colony feels slick to a fingertip, and why a colony under stress will shed sheets of mucus that hang from it in the current. Between the outer skin and the inner lining lies a layer of jelly-like material, thin in a small polyp and thick in the large solitary corals, and running through the tissues is a loose network of nerve cells with no center and no hierarchy. A touch anywhere on the polyp spreads outward through this net, and the muscle fibers in the wall and the base contract in response, hauling the whole animal down into its limestone cup until only a closed dome of tissue shows at the surface. That withdrawal is what a walker sees on a reef flat when a bright coral head dulls and shrinks a moment after a shadow crosses it. The limestone is secreted by the layer of cells on the underside of the polyp, and it is deposited outside the body, so the animal sits on and within its own skeleton rather than carrying it inside. The mineral is aragonite, one of the crystal forms of calcium carbonate, drawn from the calcium and the dissolved carbonate of ordinary seawater and laid down in fine bundles of needle-shaped crystals. Each polyp builds its own cup, with a floor, a wall, and a set of radiating vertical blades that stand up from the floor like the spokes of a wheel and press into the folds of the polyp's base. Those blades are the reason a piece of dead coral, dried and held to the light, shows a field of small starred pits, and the reason a specialist can identify a coral to species from the skeleton alone, with no soft tissue remaining. As the colony grows upward, each polyp raises itself and seals a new floor beneath, abandoning the space below so that the living animals occupy only a thin skin over a mass of vacated stone. A colony begins as one polyp and grows by budding. In some corals, a polyp constricts across the middle and becomes two, both retaining a share of the original mouth and tentacles, and in others, new polyps arise from the tissue that stretches between existing ones, spreading sideways as the skeleton spreads with them. All of the polyps in a colony are descended from that first settler and are genetically identical, and in most reef corals, they remain connected by a continuous sheet of living tissue over the surface of the skeleton, with canals running through it that link one body cavity to the next. Food taken in at one corner of a colony can therefore be shared with polyps that caught nothing, and a signal that makes one polyp withdraw can travel across the surface as a slow wave of closing mouths. That corals are animals at all was settled surprisingly late. The red coral of the Mediterranean had been fished, carved and traded for thousands of years, and the ancient explanation, repeated by Roman writers and still current in the 17th century, held that it was a plant, soft and pliant, while growing under the sea and hardened by contact with the air when raised. Naturalists of the 16th and 17th centuries placed corals in a category of stone plants, between the mineral and vegetable kingdoms, and the branching form, the fixed base and the absence of visible movement all supported the reading. When an Italian naturalist working in the early 1700s described small white flowers opening from the surface of living red coral kept in seawater, he took them as the blossoms of the plant, which was the interpretation his framework allowed. The correction came from a physician of Marseille who examined coral brought up from the Barbary coast, kept branches alive in warmed seawater, and watched the flowers open and shut. He found that they contracted when the water was disturbed, when the branch was touched and when it was moved, that they were attached by their bases to the stony branch rather than growing from a stalk, and that they behaved in every respect like the small animals then loosely called insects. He wrote up the observations and sent them to the Academy in Paris in the 1720s, where they were received with reluctance, presented without the author's name attached, and largely dismissed. He spent the rest of his career in the Antilles, and the memoir he later composed on the subject remained unpublished in his lifetime. Confirmation accumulated over the following decades from several directions at once. Experiments on the freshwater hydra in the 1740s showed that an organism with a plant-like body and a ring of tentacles caught living prey, digested it, and could be cut in pieces that each regrew into a whole, which loosened the assumption that immobility implied vegetable nature. Naturalists in France and in England examined the fine encrusting growths of the tide line, drew the polyps in their cups, and argued from their behavior and structure that these were colonies of animals. The classifications of the later 18th century still hedged, grouping corals and sponges together as intermediate creatures. But the direction of the evidence was fixed, and by the early 19th century, the coral polyp had been placed firmly among the animals whose bodies are built on a single opening and a ring of stinging tentacles, alongside the sea anemones and the jellyfish. What distinguishes the reef corals from an anemone is not much. A smaller body, a colonial habit, and the patient deposition, night after night, of a floor of limestone. The living tissue of a shallow coral is rarely white, and rarely the color of stone. It is fawn, olive, cinnamon, mustard, chocolate, sometimes a dull green, and the pigment responsible for most of that warmth does not belong to the animal at all. When 19th century microscopists began to cut thin sections of anemones, corals, and other soft-bodied sea creatures and hold them up to the light, they found the inner tissue layer crowded with small round bodies of a yellowish brown, each about the size of a red blood cell, packed so densely that they gave the whole preparation its tint. At first, they were taken for granules of the animal's own making, pigment stores or products of digestion, but they held their round shape, they had a wall, and in some preparations they could be seen to divide. The name eventually attached to them was compounded from Greek words meaning animal and yellow cell, and it stuck for more than a century as a convenient label for a whole assortment of golden cells found inside marine hosts. The same bodies turned up in sea anemones, in the mantle tissue of giant clams, in certain jellyfish, in single-celled radiolarians drifting in open water, and in the shells of some foraminifera. Chlorophyll was detected in them. Starch-like grains appeared inside them. By the closing decades of the 19th century, the reasonable conclusion had been drawn that these were plants of some kind living inside animal tissue, and that a great many of the conspicuous invertebrates of warm, shallow seas were in effect double organisms. What they are, in modern terms, is dinoflagellates, a group of single-celled algae that ordinarily swim with two whip-like flagella, one trailing and one wrapped in a groove around the middle of the cell. Inside coral tissue, they abandon that swimming form and settle into a rounded state without flagella, roughly 10 micrometers across, and each one sits within a pocket of membrane inside a single cell of the coral's inner lining, the layer that faces the gut cavity. A square centimeter of coral tissue may hold something on the order of 1 million of them, and in a large colony, the total number runs far beyond counting, distributed through the thin living veneer that covers the limestone. For a long time, the nature of the arrangement remained open. The algae might be parasites, taking shelter and nutrients and giving nothing back. They might be a captive food supply, cultivated and periodically digested, which some early writers suspected because degraded algal cells could be found in the host's tissue. They might be an accident of no consequence, tolerated because they did no harm. Deciding between these possibilities required not more looking but measuring, and the measuring began with oxygen. The experiments were simple in outline. A piece of coral was sealed in a vessel of seawater, and the dissolved oxygen was measured before and after a period of light, then before and after an equal period of darkness. In darkness, the coral consumed oxygen, as any animal does. In light, the water gained oxygen, sometimes a great deal of it, which meant that photosynthesis inside the tissue was not merely occurring, but running faster than the whole double organism could respire. Corals in bright, shallow water were found to release oxygen through the middle of the day, and the reef flat as a whole was shown to swing between oxygen surplus by afternoon and depletion before dawn. That established production. It did not yet establish whether the animal received any of the product. The answer came with isotope tracers in the middle of the 20th century. Seawater was labeled with radioactive carbon in the form of bicarbonate. Corals were left in the light for a period of hours, and the tissue was then separated, the algal cells from the animal cells, and each fraction measured for radioactivity. The label appeared first in the algae, as expected, and then, within hours, in the animal tissue and in the mucus the coral secretes over its surface. Careful chemical work on the compounds carrying the label identified the substances passing across. Glycerol in quantity, glucose, simple organic acids, amino acids and lipid material. The transfer was not a matter of the coral digesting its lodgers, since the intact algae released these compounds while still alive, and something in the surrounding host tissue appeared to prompt that release. Because algae isolated in plain seawater kept most of their photosynthetic product to themselves. From those measurements came estimates of how much of a coral's daily energy comes from within. In well-lit shallow water, the greater part of it does, and for many species, the algae can cover the whole of the animal's respiratory requirement, with a surplus left over for growth and mucus and reproduction. What the algae cannot supply is nitrogen and phosphorus in any great abundance, and this is why a coral that is fed on sunlight all day still opens its tentacles at night and takes plankton, and why colonies deprived of prey grow slowly even in bright light. The partnership is not a substitute for feeding, it is a subsidy laid on top of it. The flow in the other direction is easy to overlook and just as important. The animal respires, and the carbon dioxide it produces is delivered directly to cells that need it, at a concentration far above that of the surrounding sea. The animal excretes ammonium, and rather than losing it to the water, the coral's algae take it up and build it into amino acids that partly return to the host. Phosphate follows the same short circuit. The consequence, worked out over decades of chemical measurement on reef flats and in flowing seawater tanks, is that a coral colony holds on to scarce nutrients instead of leaking them, and that a reef can carry an enormous standing crop of living tissue in water so clear and so poor in dissolved nutrients that the open ocean around it supports very little at all. The clarity that makes tropical water look empty is a consequence of that poverty, and the same clarity lets the light down to the corals that have arranged to do without. Light also enters the arithmetic of the limestone. Corals deposit calcium carbonate considerably faster in the light than in the dark, by a factor that varies with species and circumstance, but is consistently large, and colonies whose algae have been lost or suppressed slow their skeletal growth sharply. The precise chain of cause remains a matter of active work, involving the removal of carbon dioxide, the supply of energy for pumping ions, and the production of the organic matrix into which the crystals are laid. The practical outcome is not in doubt. Reef building at any speed is confined to water shallow and clear enough for photosynthesis, which in most places means the upper tens of meters, and the shapes of colonies follow the light, branching and mounding where it is strong, flattening into plates and tables where it is scarce, tilting their surfaces toward the brightest quarter of the sky. The dependence has a visible failure mode. When water grows too warm for too long, particularly in still, bright conditions, the partnership breaks down. The algae are expelled or degraded or lost from the tissue, and the coral's own tissue, being nearly transparent, allows the white skeleton beneath to show through. A bleached coral is not dead. It is starving, living on stored reserves and on whatever it can catch, and if the water cools within a few weeks, it may repopulate its tissues from surviving cells and from the sea and regain its color over months. If the heat persists, it does not. The label that once covered all these golden cells has since been taken apart. Genetic methods applied from the 1990s onward showed that the algae in corals are not one species, but many distinct lineages, differing in the temperatures and light levels they tolerate, in how much carbon they hand over, and in which hosts they occupy, and the group has since been divided into several genera. A single colony may hold more than one kind at once, in different proportions on its shaded and sunlit faces. Some corals inherit their algae directly, packaged into eggs or into brooded larvae, and set out already stocked. Others produce larvae that are entirely without them and must acquire their algae from populations living free in the water and in the sediment, taking them up through the mouth in the days after settling, so that in a large part of the reef-building corals, the arrangement is negotiated afresh in every generation. The skeleton beneath all of this is made of a mineral the coral assembles from ingredients already dissolved in seawater, and the assembly happens in a very thin space at the base of the animal. The lower surface of the polyp, the part in contact with the limestone, is a specialized sheet of cells that pumps calcium ions inward and adjusts the chemistry of the fluid trapped between cell and stone until carbonate ions become abundant enough to crystallize. And there, in a film of liquid no thicker than a sheet of tissue paper, needles of aragonite grow outward in bundles. Aragonite is one of the two common crystal forms of calcium carbonate, denser than the other and slightly less stable over long spans of geological time, and it is what nearly all reef-building corals lay down. The crystals do not accumulate as a shapeless mass. They are guided by proteins the animal secretes among them, and they build the particular architecture of the species. The cup that holds the polyp, the ring of wall around it, the thin vertical plates that radiate inward like the spokes of a wheel and give a coral head. When it is dry and held up to the light, the appearance of a field of small stone flowers. As the colony grows, the polyp does not sink deeper into its cup. It lifts, laying down thin floors beneath itself and abandoning the space below, so that the interior of a massive coral head is a stack of sealed chambers, empty and dry of tissue, with the living film only on the outermost few millimeters of the whole structure. A boulder-shaped coral the size of a car is therefore alive only as a skin. Everything within it is architecture, produced by earlier generations of the same colony and left behind, and it is that habit of walking upward on its own leavings that makes the coral a builder rather than merely a mineral-bearing animal. The speed of that building varies more between growth forms than between climates. Branching corals with slender open arms put their carbonate into extension rather than density, and the fastest of them add something like 10 to 20 centimeters of length along a branch tip in a year, which is why a broken fragment can rebuild a thicket in a decade. The heavy, rounded, dome-shaped corals grow far more slowly outward, commonly on the order of a centimeter a year. Sometimes less, but the material they lay down is compact and resists breaking, and the largest of them, several meters across, have been alive for centuries. Their slowness turned out to be useful. From the 1970s onward, when slabs cut from such colonies were photographed with X-rays, the film showed paired bands of denser and less dense skeleton, one pair for each year, in much the same way as the rings of a tree, and by counting inward from the living surface, it became possible to date each layer and to measure how much the colony had added in every year of its life. The growth of a coral colony and the growth of a reef are not the same arithmetic, and confusing them leads to figures that are far too large. A reef is not a solid mass of coral skeleton. Cut into one or drill through it, and what appears is a loose framework of coral heads and branches, many of them broken, tilted, or resting where they fell, with all the spaces between them filled by fragments, by shell, by mud, and by sand. The sand and rubble commonly make up more of the volume than the standing framework does. Some of that sediment comes from corals ground down after death, and a great deal of it comes from other organisms entirely. Calcareous green algae that grow as chains of small limy segments and shed them by the handful when they die, single-celled foraminifera with chambered shells the size of a grain of coarse sugar, the shells of snails and clams, the spines and plates of sea urchins, and the fine flour produced when a fish scrapes rock. Loose sediment alone would wash away in the first storm. Two agents hold the pile together. The first is a group of red algae that grow not as fronds but as hard pink crusts, laying down their own calcium carbonate in the form of a magnesium-rich calcite, and spreading over dead coral, over rubble, over shell, and over each other in layers that are thin but immensely tenacious. They plaster fragments to the framework, seal the surfaces of cavities, and on the windward edges of many reefs, where the surf is heaviest, they build a low ridge of their own that is more algal than coral, a pavement of pink and mauve rock lying exactly at the level of the breaking waves. The second agent is chemical. In the dark spaces inside a reef, seawater circulating through the pores becomes locally saturated and precipitates fresh carbonate directly onto the walls, as fine crusts of needles or as small radiating fans, so that over centuries the loose interior is slowly turned into rock without any organism being involved at all. Working against all of this continuously is a population of animals whose business is the removal of limestone. Certain sponges bore into coral skeleton by chemically etching out tiny chips of it, hollowing dead heads into a lattice so fine that the rock rings hollow and breaks under a hand. Bivalves settle as larvae on living coral and grind their way in, sitting for life in a flask-shaped burrow with only their siphons exposed. Worms of several unrelated kinds bore, as do certain barnacles and small crustaceans, and the surfaces are grazed constantly by urchins that rasp with a ring of teeth, and by parrotfish whose fused beaks bite off mouthfuls of rock along with the film of algae growing on it. Parrotfish digest the living part and pass the mineral, and a single large one may release several hundred kilograms of fine carbonate sand in a year, so that a good deal of the pale beach around a reef island has been through the gut of a fish. This erosion is not a flaw in the system. It supplies the sediment that fills the framework. It prunes the reef of weak and overhanging material before storms take larger pieces, and it opens the shelter that a large part of the reef's animal population lives in. What accumulates in the end is the small remainder left after the building, and the removal are set against each other, and the accounting is done as a carbonate budget. Kilograms produced per square meter per year on one side, kilograms bored, ground and washed off the edge into deep water on the other. The difference is modest. Reefs in good condition, taken as whole surfaces rather than as single fast-growing colonies, typically gain height at rates measured in a few millimeters a year, and a meter of new reef in a century is brisk work. Under that reckoning, the great structures make sense only over thousands of years, which is the span the cores have provided. There is also a ceiling. Coral cannot survive long exposure to air, so vertical growth stops at about the level of the lowest tides, and once a reef has reached that level, it can only grow sideways or upward by having storm rubble and sand heaped on it above water. This is why so many reefs share the same flat profile: a broad pavement close to the surface, a rim of the toughest growth at the seaward edge, and steep, actively growing flanks below. Height beyond that requires the sea itself to rise, and it has. After the last glacial period, the meltwater raised sea level by well over 100 meters, and reefs responded in 3 ways that have been read out of drill cores again and again. Some grew fast enough to keep their crests near the surface throughout. Some fell behind, remaining in deeper water while the rise continued, then caught up when it slowed. Others were left too deep, too turbid or too cold, and simply stopped, leaving drowned reef terraces on the slopes below the living ones. The living reefs of today therefore sit as a veneer, often only 10 to 30 meters thick and a few thousand years old, resting on far older limestone laid down during earlier high seas, and beneath that on limestone older still, the whole platform a stack of interglacial reefs separated by the weathered surfaces of the times when the sea was low and the coral was gone. A single reef, considered as a unit of study, holds more kinds of animal than any observer can hold in mind at once, and the first task of the naturalists who set out to catalogue one was not identification but simply counting, which turned out to be the harder problem. Fish could be tallied along a measured line, a diver swimming slowly with a slate and noting every individual within a few meters on either side, and repeated transects of that kind laid across the flat, the crest and the upper slope gave the first reliable lists. What the transects missed was almost everything else. The animals that live inside the reef, in the rubble, in the sand pockets, in the dead cores of living coral heads, do not swim past a diver, and to find them the reef had to be taken apart. Coral heads were lifted whole into bags, carried up, broken open on a boat deck and sorted, and small enclosed patches were sampled with anesthetics that brought the hidden fish out of their holes. The results reorganized the way the reef was understood. The large and conspicuous residents, the ones that appear in paintings and in memory, are a minority of the species present, and the greater part of the community is small, cryptic, and permanently out of sight. Even so, the visible fish are the part of the reef most thoroughly recorded, and the tally is considerable. More than 1,500 species of fish have been listed from the Great Barrier Reef as a whole, and a single well-worked reef within a rich region may hold several hundred, drawn from a set of families that recur across the tropics with different species filling the same offices. Damselfishes hover in clouds over branching coral and drop into it when a shadow passes. Wrasses of many sizes work the bottom, turning small stones, cracking snails, following the diggings of larger fish. Parrotfishes move in loose companies across the pavement, and the sound of their beaks on limestone carries through water clearly enough that observers learn to locate them by ear. Butterfly fishes travel in pairs along the reef edge with a deliberate slowness, and many of them eat nothing but coral, picking individual polyps or taking mucus from the surface. Surgeon fishes crop the low turf of algae. Groupers and moray eels wait in the deeper openings. Above the whole structure, where the current runs, ranks of small planktivores face upstream and pick particles out of the water one at a time, and their position in the flow is so consistent that a photograph of a reef edge can be used to infer the direction the tide was running. Sorting these fish by what they eat produced the concept of feeding guilds, and the guilds explain a good deal about how the reef looks. Grazers keep the open pavement short so that algae there grow as a dense, low mat rather than as fronds, and where grazing is prevented by a cage or interrupted by disease, the same surfaces turn shaggy within weeks. The excavating parrotfishes remove not just algae but the limestone beneath, which links the fish community directly to the carbonate accounting of the reef, since much of the sand in a lagoon has passed through their guts. The corallivores impose a slow tax on living tissue, visible as pale bite scars on plate corals. The predators, groupers, snappers, trumpetfish, jacks that sweep in from open water remove a share of the small fish each day, and the risk they impose is why so many reef fish spend their lives within a metre or two of shelter. Home ranges measured for the smaller gobies and blennies can be a matter of tens of centimetres. An entire life spent on one coral head, and the fish that seem to be drifting idly over a thicket are in fact holding station at a fixed distance from a hole they can reach in less than a second. Some of these residents defend space with a persistence out of proportion to their size. The territorial damselfishes maintain patches of algae on dead coral and treat them as holdings, chasing away surgeon fishes and parrotfishes many times their length, and inspection of those patches showed something more particular than simple exclusion. The algae inside a defended territory differ in species composition from the algae outside, because the damselfish removes the kinds it does not favor and tolerates the kinds it eats. Naturalists took to calling the patches gardens, and the word is nearly accurate since weeding is the operative activity. The consequences extend to the coral, as the algal mats trap sediment and can kill the colonies they grow on, so that a dense population of these fish leaves an identifiable mosaic on the reef surface. The arrangement that most surprised early divers was cleaning. Certain small wrasses, banded in blue and black, occupy fixed positions on the reef, and other fish come to them, stop, hold themselves at odd angles with fins spread and gill covers lifted, and permit the small fish to move over their skin, into the mouth and among the gill filaments, removing parasites and loose tissue. The behavior was described in the scientific literature from the 1950s onward. Once air supplied from a cylinder allowed an observer to stay in one place long enough to watch a station operate through the course of a day. And the counts made at such stations run to hundreds of clients in a few hours, including large predators that would otherwise eat a fish of that size. Cleaner shrimps do the same work on the ceilings of caves, waving long white antennae. There is also a mimic, a blenny patterned almost exactly like the cleaner rasa, and swimming with the same forward and back motion, which uses the resemblance to approach and then takes a bite out of a fin and retreats. And the existence of that deception is part of what convinced observers that the cleaning relationship is a real and dependable feature of reef life rather than a chance encounter given a name. The mollusks are less easily watched, but no less numerous. Giant clams sit wedged into the reef flat with their fluted shells gaping and their mantles turned to the light, colored brown and blue and green by the same algae that live inside coral tissue, so that the clam feeds partly by farming. Cowries and cone shells emerge at night, the cones hunting worms, other snails or small fish, which they take with a hollow harpoon tooth loaded with venom. Nudibranchs, shell-less and often vividly patterned, browse on sponges and hydroids, and some of them swallow the stinging cells of their prey undischarged and mount them along their own backs. Octopuses keep dens marked by heaps of emptied shells, which is how their diet was first worked out, by sorting middens rather than by watching the hunt. Boring bivalves live sealed in the limestone with only their siphons showing, and the tubes of Christmas tree worms open into the surface of massive corals as small paired spirals that vanish instantly when a hand comes near. Crustaceans occupy nearly every crevice. Snapping shrimps produce the crackling that fills any hydrophone recording of a reef, a sound made by the collapse of a cavitation bubble when the enlarged claw closes, and many of them share their burrows with a goby that stands at the entrance and signals with its tail when danger approaches, the shrimp keeping one antenna in contact with the fish while it works. Small crabs live permanently among the branches of certain corals, feeding on mucus and defending the colony by pinching the tube feet of starfish that try to climb aboard. Mantis shrimps hold burrows in the rubble and strike with limbs that either spear or shatter, and their eyes, examined in detail, prove to have more classes of color receptor than any other animal known. The echinoderms complete the account. Sea urchins wedge themselves into hollows by day and graze the pavement at night, and their contribution to keeping algae short was demonstrated involuntarily when a disease swept through the Caribbean in the 1980s, killing the common long-spined urchin over an enormous area and leaving reefs that turned from limestone to weed within a year or two. Brittle stars fill the interior spaces, hundreds to a single coral head extending arms into the water after dark. Sea cucumbers move over the sand, ingesting it, passing it through and depositing it cleaned. Feather stars climb to exposed corners of the reef edge at dusk and open their arms into the current, and the crown of thorns starfish, when present in numbers, feeds on the coral itself by turning its stomach out over the colony and digesting the tissue where it lies. The change begins before the light has properly gone, in the half hour when the water above the reef turns from blue to grey and the far edge of the drop-off becomes indistinct. And it is one of the few transitions on a reef that a person can watch from beginning to end without moving. Divers who have timed it describe a period of unusual traffic, when the fish of the day are hurrying inward toward the coral, and the fish of the night have not yet come out, and then a brief interval in which the reef looks nearly empty. The open water above the pavement holding almost nothing, before the second population appears from the same holes the first has just entered. The whole exchange, from the first departures to the settling of the night arrangement, takes somewhere between 20 minutes and an hour, depending on the depth, the clarity of the water and the phase of the moon. The daytime fish do not simply drift into shelter. They return to particular places, and many of them go back to the same crevice night after night. Surgeon fish and butterfly fish press themselves into gaps barely wider than their bodies, wedging in with the dorsal fin raised so that a hand cannot easily draw them out. Damselfish sink down into the branches of the coral they have defended all day and lie among them at odd angles. Wrasses of several kinds bury themselves in sand, disappearing entirely, and can be found by turning the sediment over with a fin. Certain parrotfishes secrete a covering of mucus from glands near the gills and lie inside it, a loose transparent envelope with an opening at either end, and experiments in which the envelope was removed suggested it interferes with small parasitic crustaceans that swim up from the sediment at night to feed on fish blood. A number of fish also change color as they settle, replacing the patterns they wore in daylight with blotches and bars that break up the outline against rock. The interval of dim light is the most dangerous part of the night for a small fish, and this has been measured rather than merely observed. Jacks, trevallies, snappers, groupers, and coral trout hunt hardest at dusk, and again at first light, when there is enough light for a predator looking upward to see a silhouette against the surface, but not enough for the prey, looking down into a dark reef to see the predator coming. White-tip reef sharks that have spent the day lying under ledges become active, moving into the coral and pushing their heads into holes, and the sound of a hunt at night carries as a series of scrapes and thumps, rather than as anything visible. The fish that emerge have large eyes set high in the head, and many of them are red, a color that appears nearly black in water, where the red end of the spectrum has already been absorbed. Squirrelfish and soldierfish hang under overhangs by day and come out over the pavement after dark. Big eyes and cardinal fish rise into the water column to pick at drifting animals, and moray eels leave their holes entirely and travel across the reef with their bodies threaded through the coral. Behind the retina, many nocturnal reef fish carry a reflective layer that returns light through the light-sensitive cells a second time, which is why their eyes shine back at a torch. Grunts and certain snappers make longer journeys, forming tight schools over the reef during the day and then dispersing at dusk across the sand flats and seagrass beds to feed, returning before dawn along routes so consistent that they can be predicted, and in doing so they carry nitrogen and phosphorus from the flats back to the coral in their waste, which has been measured as a meaningful input to the reefs where they gather. Meanwhile, the water itself changes. Through the day, a great part of the small animal life of the open ocean stays well below the surface, and after sunset it rises, a movement repeated across the world's oceans and first mapped in detail with echo sounders, which returned an echo from a layer that appeared to be the seabed at a few hundred meters, and then rose toward the surface each evening and sank again at dawn. The layer proved to be animals, mostly small crustaceans and fish and squid, moving up to feed in darkness and down again to avoid being seen. On reefs, the same pattern brings copepods, arrow worms, larval fish, larval crabs and swimming mollusks into the water above the coral, and a plankton net towed at night over a reef collects several times the mass it collects over the same ground at noon. To this is added a population that comes from the reef itself, small crustaceans that spend the day within the sand and the rubble and swim up after dark, which explains why light traps set on the bottom fill so quickly. The corals respond to this arrival. Many reef-building corals keep their polyps contracted through the day when the algae in their tissue are working, and expand them at night so that a colony that looked like smooth stone at noon becomes furred and soft to the eye, each polyp holding its ring of tentacles open into the current. The stinging cells fire on contact, and the tentacles fold inward to carry what they catch to the mouth, and stomach contents examined under a microscope have shown copepods and other small crustaceans in quantity. Some corals feed instead by spreading sheets and threads of mucus over themselves and drawing them back in, loaded with fine particles. Certain species also extrude their digestive filaments after dark, laying them over the tissue of a neighboring colony that has grown too close and digesting it where it touches, and where two species meet regularly. One of them may grow elongated tentacles reserved for this contest. Calcification, measured through the day and night, generally runs faster in daylight when photosynthesis is underway, and the alternation of fast and slow deposition leaves fine banding in the skeleton. The invertebrates of the reef surface are largely nocturnal, and after dark the pavement becomes crowded. Feather stars that spent the day folded in crevices climb to exposed edges and open their arms. Brittle stars extend a few arms at a time from inside coral heads. Basket stars unroll a mass of finely branched arms into the current, forming a mesh that is drawn in and cleaned at intervals. Sea urchins move out and graze the rock, cutting into the limestone as they go and leaving pale scars. Cowries emerge and spread the folds of the mantle back over their own shells, which is why the shells are polished. Cone snails hunt along the sand with the siphon raised. Spiny lobsters leave their dens and walk out over open ground, sometimes in single file, and return before daylight to the same shelters, which have been mapped by tagging. Light of another kind appears on the darkest nights. Single-celled dinoflagellates in the water flash when the water around them is sheared, so that a hand drawn through it leaves a trail of blue-green sparks, and a wave breaking on the reef crest can outline itself. Small crustaceans called ostracods in parts of the Caribbean release luminous material in short pulses as they swim, producing patterned displays close to the reef surface after sunset. Flashlight fishes carry pouches beneath the eyes packed with luminous bacteria, kept permanently and supplied by the fish, and they can cover and uncover the light, appearing in numbers near reef walls on moonless nights and staying away when the moon is full. In deeper water, various soft corals and other animals produce light of their own when touched. The reef is also noisier at night than by day, though the sound is different in character. The scraping of parrotfish teeth largely stops, the crackle of snapping shrimps rises, and various fishes produce grunts, knocks and drumming that build into choruses at dusk and again before dawn, patterns clear enough on a hydrophone record that a listener can tell the hour from them. Recordings played back in the open sea have been shown to attract the larvae of reef fish and invertebrates, which suggests the nightly sound of a reef is one of the signals by which the young of the reef find their way back to hard ground. Toward the end of the night, the water over a shallow reef flat holds less oxygen than at any other time, because the algae and the corals have been respiring for hours without photosynthesizing, and measurements on enclosed flats and lagoons show a steady decline through the dark that reverses within minutes of sunrise. As the light returns, the whole exchange runs backwards, the nocturnal fish withdrawing into their holes, the polyps drawing in their tentacles until the colonies look like stone again, the urchins retreating, the parrotfish leaving their mucus envelopes hanging empty in the coral, and the daytime population coming out into a reef that shows almost no trace of the night's work. On a small number of nights each year, the ordinary rhythm of the reef is interrupted by something that runs on a longer clock, and for an hour or two, the corals themselves become the most active thing in the water. The colonies that have spent the year adding limestone in millimeters release their eggs and sperm into the sea at almost the same time. Whole hillsides of coral emptying within a window narrow enough to be measured in minutes, and the water above them fills with buoyant pink and cream and pale orange packets that rise slowly toward the surface. Divers who have watched it describe the effect as a rain falling upward, and it is one of the few events on a reef that can be predicted to the night and very nearly to the hour. For a long time, this was not known. Naturalists working in the 18th and 19th centuries had found small swimming larvae inside certain corals, and understood that at least some species produced young that were brooded within the parent and released ready to crawl. And from this it was widely assumed that corals reproduced quietly and continuously, a few larvae at a time, without any spectacle. Reports of oily slicks and a smell of spawn on the sea after full moons circulated among fishermen and coastal observers, without being attached to any particular animal. The synchronous release of gametes by many species at once on tropical reefs was documented in detail only in the early 1980s by researchers diving at night on the Great Barrier Reef with lights and collecting nets. And once described there, it was looked for and found on reefs in other regions, each with its own season and its own set of participating species. What the divers saw was that the corals do not simply leak their gametes, but package them. In the hours before release, many of the branching and massive stony corals bring bundles of eggs and sperm up into the mouth of each polyp, where they sit visible through the tissue, a stage that observers call setting because it gives an unmistakable warning that the colony will spawn within a short time. When the moment comes, the polyp opens and the bundles are pushed out, and because they contain a good deal of lipid, they float, drifting up through the water column and gathering at the surface in windrows and rafts that can extend for a long way downwind. There the bundles break apart, the sperm swim free, and the eggs are fertilized in the thin, warm layer at the top of the sea, so that the reproduction of an animal fixed permanently to the bottom happens in the last few centimeters of the water above it. The timing appears to be set by at least 3 separate signals working at different scales. Rising sea temperature through the spring or summer determines the month, so that on the Great Barrier Reef, the main spawning falls in the warming months of the southern spring, while in the Caribbean it comes in late summer, and cooler outer reefs may run behind warmer inshore ones by several weeks. The phase of the moon determines the night, with most species releasing on particular nights following a full moon, when the dark interval between sunset and moonrise is longest, and the fading of daylight determines the hour, since colonies held in tanks can be shifted earlier or later by changing the light regime. Corals possess light-sensitive proteins of the cryptochrome family, whose activity in some species varies with the lunar cycle, and these are the best current candidates for the mechanism by which a coral reads moonlight and counts the nights. Within a single night, the species do not all go at once, and the ordering is consistent enough that experienced observers can name what is spawning from the clock alone. Some corals release within half an hour of dark, others 2 or 3 hours later, and closely related species that could otherwise interbreed are often separated by these staggered windows, so that their eggs meet mostly the sperm of their own kind. Many stony corals are hermaphrodites, each polyp making both eggs and sperm, and would seem to risk fertilizing themselves, but in most species the eggs are chemically incompatible with sperm from the same colony, and the bundles must find another colony's contribution to develop. Because a coral colony and its neighbors may all be clones grown from broken fragments, this incompatibility can extend across a whole patch of reef, which makes the wide mixing at the surface useful rather than merely incidental. Fertilization in open water is a matter of dilution, and the synchrony compensates for it. Sperm are diluted very quickly by mixing, and measurements at spawning slicks show that fertilization rates are high in the first minutes and fall away sharply as the gametes spread, which is why calm nights with little wind and slack tidal flow tend to produce the most successful spawning, and why the whole reef going at once matters more than any individual colony's contribution. The slicks themselves attract attention. Fish rise into them to feed, plankton-eating birds work them by day if any remain, and a heavy spawning in an enclosed lagoon can consume so much oxygen as the excess material decays that the water turns milky and animals in it come under stress. The fertilized egg divides into a flattened ball of cells that is fragile and easily broken by rough water, and within a day or so it has become a small ciliated swimmer called a planula, elongated without a mouth at first, propelled by beating cilia and carrying the lipid reserve packed into the egg as its only food supply. Species that brood their young arrive at a similar stage by a different route, taking sperm in from the water, fertilizing eggs inside the polyp, and releasing larvae that are larger, further developed, and often already carrying the symbiotic algae inherited from the parent. Brooded larvae tend to be competent to settle almost immediately and to travel short distances. while the larvae from broadcast spawning generally acquire their algae only after settling from the water and the sediment around them and spend longer adrift. How far they go is decided mostly by the currents, since a planula swims slowly and can do little against a moving body of water, but it is not entirely passive. Larvae adjust their depth, and by moving up or down, they can shift between layers of water traveling in different directions or at different speeds, which changes the outcome of a drift considerably. Most planulae become able to settle within a few days and lose that ability after some weeks, though a few have been kept alive and competent in tanks for a couple of months, and genetic comparisons between reef populations suggest a pattern in which the large majority of recruits settle within a few kilometers of where they were spawned, while a small minority cross open water and link reefs that lie far apart. Settlement is selective. A competent larva swims down, tests surfaces with the end away from its mouth, and responds to chemical cues from the crusts of coralline algae that coat clean hard rock, to the bacterial films that develop on such surfaces, and apparently also to the ambient sound and the color and texture of the substrate, favoring pale coralline pink over bare stone and often choosing crevices, pits, and the undersides of ledges where a young polyp is less likely to be grazed away or buried. Once it has chosen, the larva attaches, flattens into a disc, and secretes the first thin plate of aragonite beneath itself, then opens a mouth, grows a ring of tentacles, and becomes a primary polyp perhaps a millimetre across, at which point it begins the budding that will make a colony. The arithmetic that follows is severe. A large colony may release many hundreds of thousands of eggs in a night, and of those a small fraction are fertilized, a small fraction of the embryos survive to swim, a small fraction of the swimmers find suitable ground, and a small fraction of the settlers survive their first months against grazing fish, shifting sand, competing algae, and the simple accident of being in the wrong centimetre of reef. Plates of terracotta and settlement tiles left out on reefs and collected months later have allowed researchers to count recruits directly, and the counts are low, a few young colonies on a hand-sized plate in a good year, and none at all in a poor one, which is the ordinary rate at which the structures that took thousands of years to build are replenished. Reefs were known intimately long before they were written about. People living on coasts and low islands within the tropics had names for particular passages and particular heads of coral, knew which channel could be crossed on which tide, quarried reef rock for walls and graves and lime, read the color of water as a chart, and carried that knowledge in speech and in practice rather than in print. The written record that accumulated in European libraries began later and began with hazard, in sailing directions that warned of pale green water and broken swell, in log entries that recorded a bottom felt suddenly by the lead where an hour earlier there had been no bottom at all, and in the standing instruction that a man should be sent to the masthead in the middle of the day, when the sun was high enough to show a shoal as a stain on the sea. The most consequential of those encounters for the history of the subject took place in 1770, when the Endeavour under James Cook, working north along the eastern coast of what is now Queensland, struck a reef at night and hung there with water rising in the hold. The ship was lightened, hauled off, plugged with a sail drawn under the hull, and brought into a river mouth on the mainland to be beached and repaired. And during the weeks of that repair, the the naturalists aboard, Joseph Banks and Daniel Solander, collected and described what lay around them. Afterwards, the ship had to feel its way north inside a labyrinth of shoals, sounding continually, sending boats ahead, and eventually finding a gap through the outer reefs into open sea, which gave Cook occasion to describe in his journal a wall of coral rock standing against the ocean swell with deep water immediately outside it. What came home from such voyages was less than what had been seen. Coral collected alive and dried in the sun kept its shape and lost almost everything else, so the cabinets of Europe filled with white and pale branches, plates and boulders, stripped of tissue, stripped of color, stripped of the extended tentacles that would have settled the question of what kind of thing they were. Specimens preserved in spirits of wine fared a little better, though the polyps contracted into knots and the pigments leached into the fluid. Reef fish shrank and faded in their jars. The result was a body of evidence that consistently understated the living reef and encouraged the older view that these were mineral growths, stone vegetation, something intermediate between rock and plant, and it took the drawings made on the spot to correct the impression. Voyage artists therefore mattered a great deal. They worked in cramped quarters, in heat, with specimens spoiling in front of them, and the good ones drew the animal while it was still expanded in a bowl of seawater, recording the number and arrangement of the tentacles and the colors that had be gone by morning, sometimes with the aid of a camera lucida, which threw an image of the object onto the paper so the outline could be traced at true proportion. Sidney Parkinson, who drew for Banks on the Endeavour, died on the voyage home, and much of the material from that expedition was published slowly or not at all, which was the ordinary fate of natural history collections. Still, over several decades, the accumulated plates of expanded polyps, madrepore branches, giant clams, sea slugs and reef fish established a visual vocabulary for a kind of place that most of the readers would never see. The deeper puzzle was one of form rather than biology, and it presented itself most sharply in the open Pacific. Ships crossing from South America toward Asia passed low rings of sand and palm enclosing shallow lagoons, with no mountain, no hill, no rock other than coral limestone, standing in water so deep that soundings a short distance offshore found no bottom at all. The naturalists on Cook's second voyage, Johann Reinhold Forster and his son Georg, saw such islands and wrote about them, and the question they and others raised was how animals which lived only in shallow, clear, sunlit water could have produced structures rising from an abyssal floor. Some proposed that corals had built upward from the deep sea over unimaginable spans. Others suspected foundations that could not be seen. Two lines of evidence narrowed the possibilities. Matthew Flinders, surveying the Australian coast in the Investigator early in the 1800s, examined the outer reefs closely, noted that the living coral was confined to a shallow surface layer while the mass beneath was dead limestone, and reasoned that a reef grows upward until it reaches the level at which the animals can no longer live, and then can grow only outward. Rather later, French naturalists working in the Pacific, among them Jean-René Constant-Coy and Joseph-Paul Gaimard, argued from their own dredging and observation that reef-building corals were restricted to modest depths, perhaps a few tens of fathoms, and could not have begun their work on the floor of the ocean. If the animals were shallow and the water was deep, then the base of every atoll had to be something else, and something else that had once been near the surface. Charles Lyell, in his Principles of Geology, offered the explanation that seemed most natural to a generation impressed by volcanoes in the Pacific, proposing that atolls were coral encrustations upon the circular rims of submerged volcanic craters, which would account for the ring and for the lagoon in the middle. The objection to it was arithmetical rather than theoretical. Atolls came in many sizes, including lagoons scores of kilometers across, far wider than any known crater, and they were not all circular, some being elongated, some bent, some enclosing lagoons of irregular outline that looked less like a crater than like a drowned coastline. Charles Darwin arrived at a different account while sailing in the Beagle between 1831 and 1836, and he arrived at it, by his own statement, on the west coast of South America, before he had seen an atoll. What he had seen there were raised beaches and marine shells lying well above the sea, evidence that large tracts of land could rise, and it followed that other tracts could sink. Suppose an island fringed by coral began slowly to subside. The corals, growing upward at the reef edge, where water was clearest and waves brought food, would keep their surface at sea level while the land behind them went down. So the fringing reef would become separated from the shrinking shore by a widening channel and would stand as a barrier reef. Let the subsidence continue until the last peak disappeared, and what remained would be a ring of coral enclosing a lagoon, with nothing inside it but water and sediment. The scheme required only two slow processes, neither of them extraordinary, and it accounted for the sequence of forms as stages of one history rather than as separate kinds of object. Darwin saw the Keeling or Cocos Atoll in the Indian Ocean in 1836 and spent his days there examining the reef flat, the ramparts of broken coral thrown up by storms, the boulders lodged on the reef top, the fine sediment of the lagoon floor and the outer slope, where soundings taken with a lead armed with tallow came up marked by living coral down to a certain depth and smooth below it. He published the argument in 1842 in a book on the structure and distribution of coral reefs, accompanied by a world map on which fringing reefs, barrier reefs and atolls were colored differently, so that the distribution itself became an argument about which parts of the ocean floor had been sinking. The theory was adopted quickly by some and resisted for decades by others. James Dwight Dana, who had seen many Pacific islands as geologist to the United States exploring expedition, supported it and added the observation that islands with barrier reefs often had deeply embayed coasts and valleys that ran out under the sea, exactly as drowned land should. Others, including Carl Semper working in Palau and John Murray of the Challenger expedition, held that submarine banks could be raised toward the surface by the steady accumulation of sediment and the remains of shelled organisms, and that reefs might then cap them and hollow out lagoons by solution and erosion, requiring no subsidence at all. Alexander Agassiz spent years and much of his own money visiting reefs in several oceans to test the matter by inspection. The dispute could not be settled from the surface, since it turned on what lay beneath 1,000 feet of limestone, and its resolution waited on the slow, awkward business of drilling into a reef and reading the cores. The lead line was the first instrument to touch a reef in any systematic way, and it remained the standard for centuries because it was cheap, robust, and understood by every seaman aboard. A shaped lead weight of 7 or 14 pounds hung from a long line, marked at intervals with strips of leather, pieces of coloured cloth and knotted cord, so that a leadsman standing in the chains could read a depth by feel in darkness as easily as by eye in daylight. The base of the lead was hollowed and filled with tallow, and this arming picked up whatever it struck, so that a sounding returned not only a number, but a sample, a smear of grey mud, or a few grains of white sand, or the fine cream-coloured powder of ground coral. Over a reef, the instrument had its awkwardness. The bottom was hard and irregular. The lead bounced or wedged in a crevice. The tallow came up scratched and empty, and the line, streaming in a tidal current across a wall of living limestone, chafed and parted. Surveyors learned to work from small boats at slack water, dropping the lead again and again along lines fixed by angles taken to marks ashore, accumulating depths one at a time. What emerged from thousands of such soundings, laboriously plotted, was the general shape of the structures. Outer slopes were steep, in many places steeper than any land hillside, falling from the surf into deep water within a short horizontal distance. Living coral was brought up, or felt on the lead, down to a few tens of fathoms and no deeper, while below that the bottom gave back sand and rubble derived from above. Lagoons, by contrast, were shallow and remarkably flat, floored with fine sediment, interrupted by isolated coral pinnacles that rose almost to the surface and that ships learned to fear more than the rim itself. Passes through the rim were often on the leeward side, and the depths in them followed the shape of channels. All of this could be mapped in plan and profile, and none of it answered the question of what lay under the limestone, because a lead line measures only the top of a thing. Charting proper, the making of a usable chart, added the geometry that soundings alone lacked. Surveyors landed on the sand cays, cleared a station, measured a baseline with a chain across the reef flat, and worked outward by theodolite and plane table, taking angles to beacons and boats, and to the conspicuous heads of coral. Tide poles were set up and read through a full lunar cycle, so that soundings taken at different hours could be reduced to a common datum, usually a level near the lowest tides, since the purpose of the chart was to warn ships rather than to describe the reef. Those charts still carry the marks of their making in the scattered pattern of spot depths that thins away from the boat tracks, and in the blank spaces where no line was run. A century of such work produced outlines accurate enough for navigation while leaving the interiors of large reef complexes almost unexamined. The interior question required drilling, and the first serious attempt was organized by the Royal Society in a series of expeditions to Funafuti Atoll, in what was then the Ellis Islands and is now Tuvalu, in 1896, 1897, and 1898. The first season failed to get far since the machinery was light. The coral rock was full of cavities into which the drilling water simply drained away, and the hole kept collapsing. Heavier casing and more patience in the following seasons drove the boring deeper, and it eventually passed 1,100 feet, something over 300 meters, still in material of reef origin the whole way down. The cores were shipped home in boxes and studied for years. They showed coral limestone, shell and sand, much of it altered, some of it converted to the harder mineral dolomite by the passage of magnesium-bearing water through the rock. The result was consistent with a reef that had grown upward as its foundation sank, but it was not a proof, because the drill had never reached a floor, and those who preferred a shallow bank as the starting point could argue that the bank simply lay lower still. The proof came half a century later from drilling on atolls in the northern Marshall Islands, undertaken alongside military work there. On Eniwetok in the early 1950s, boreholes went down through more than 4,000 feet of limestone, about 1,300 meters, and ended in basalt, the rock of an old volcano. The fossils in the limestone just above the contact were of Eocene age, so the atoll had been growing at the surface, and its foundation sinking beneath it through tens of millions of years, at rates close to those the subsidence theory required. The cores also carried something the argument had not anticipated. At intervals within the limestone, there were surfaces of solution, hardened crusts and cavities of the kind that form when limestone stands in air and rainwater rather than under the sea, recording the times when the ocean fell away during glacial periods and the reef top became a low island of weathering rock, to be flooded and colonized again when the ice melted. While drills worked from platforms above, other observers were going down to look. The standard diving dress of copper helmet and canvas suit. Air supplied through a hose from a pump on deck had been in industrial use since the early 19th century, and a simplified version, an open helmet resting on the shoulders and fed by a hand pump, was light enough to be carried in a dinghy and used in a few fathoms of water. Naturalists on the Great Barrier Reef expedition of 1928 and 1929, based for a year at Low Isles off the Queensland coast, used such gear to walk on the reef below the level that wading could reach, and so to see the zones of coral growth from the side rather than from above, to mark out plots with stakes and revisit them, and to watch corals feeding in place. Growth was measured by weighing marked colonies, by photographing staked quadrats at intervals, and later by dosing living corals with a dye that leaves a colored line in the skeleton at a known date, so that the following year's addition could be read off directly. The self-contained demand regulator, which allowed a diver to carry compressed air on the back and breathe it at ambient pressure without a hose to the surface, came into general use after the Second World War, and reef survey changed character within a decade. A diver with a mask, fins, and a tank could swim a measured line along a slope, count what lay under a tape, collect small animals from crevices, and repeat the whole exercise on the same line a year later. Photography followed, first single frames in housings, then long overlapping series joined into mosaics of the seabed, which allowed the same square of reef to be compared with itself across decades. From the air, the reefs appeared in plan for the first time. Aerial photographs taken through clear, shallow water show the zones as bands of color and texture. The surf line, the pavement, the coral heads, the sand sheets, and the sea grass. And their boundaries can be traced continuously for kilometers in a way that no boat survey could match. Blue and green light penetrates water furthest, so shallow detail records well, while depth information fades out below a few tens of meters in the clearest conditions and much sooner in turbid water. Wartime reconnaissance produced large photographic coverage of Pacific atolls for the practical purpose of finding landing places over reef flats, and much of that imagery was afterwards used by geologists. Sound completed the picture. The echo sounder, which measures the time a pulse takes to travel to the bottom and back, came into shipboard use in the 1920s and drew a continuous profile instead of isolated dots, revealing submerged terraces on reef slopes at consistent depths, drowned reef surfaces well below the living zone, and flat-topped submarine mountains in the deep Pacific whose summits lay hundreds of meters down. Later instruments sent out many beams at once and mapped whole swathes of seabed in a single pass, while side-scanning sonar returned pictures of texture, and low-frequency profilers pushed sound into the rock itself to show the layers, cavities, and buried weathering surfaces inside a reef without drilling it. Aircraft carrying pulsed green lasers now measure shallow depths by timing the reflection from the surface against the reflection from the bottom, and X-radiographs of sawn coral slabs show paired annual bands of denser and lighter limestone, so that the same structure can be read at the scale of an ocean basin and at the scale of a single year. The corals of the shallow tropics are so closely tied to sunlight that for a long time the word coral carried the sun with it, and the discovery that stony corals also live in cold water, in permanent darkness, at depths where no plant can grow, arrived piecemeal over 2 centuries and by the least glamorous of means, which is to say a weighted iron frame dragged along the bottom behind a slow-moving vessel. Fishermen in northern Europe had known for generations that certain grounds were coral grounds, hard patches where lines fouled and nets came up torn, and where the broken white branches lifted from the seabed were sometimes brought ashore as curiosities. Naturalists in the 18th century described such branching stony corals from northern specimens without being able to say much about the places they came from, and the sense that these were the debris of something larger, some structure standing on the bottom in water far too deep and too cold for the reefs of the atlases, took much longer to form. The deep dredging campaigns of the 19th century did most of the early work. Norwegian naturalists working the fjords and the shelf edge brought up stony corals, sea fans and sponges from hundreds of meters, and British expeditions in the late 1860s, sounding and dredging along the slopes west of Ireland and Scotland, recovered coral fragments together with the animals living among them. The Challenger expedition of the 1870s, which circled the world sampling the deep bottom, added coral records from many oceans and from great depths, and the specimen jars and mounted branches that came home were catalogued, named and drawn with care. What the dredge could not give was arrangement. A dredge tells you that particular species occupied a particular stretch of seabed, and it will occasionally give a hint of abundance, but it cannot show whether the corals stood as isolated bushes on a plain of mud or as a continuous thicket, nor whether the dead material beneath them formed a mound, and for the better part of a century that question stayed open. The corals themselves are built on a different economy from their tropical relatives. They carry no symbiotic algae in their tissues, so they take no share of sugars from photosynthesis and depend entirely on capturing food, which means zooplankton drifting past, the small crustaceans that migrate up and down the water column, and the flakes of organic material sinking from the productive layers far above. Their polyps are generally larger than those of shallow reef corals. Their tentacles are long and translucent, and because they have no need to house algae or to protect them from ultraviolet light, their colors are their own, white and cream most often, sometimes pink, orange, or pale yellow, and those colors survive being brought into the light. Not needing sun, they are limited instead by 3 other things: hard ground to settle on, water cold enough but not too cold, roughly between 4 and 12 degrees Celsius, for the commonest reef-forming species of the North Atlantic, and above all, a steady supply of moving water, since food must be brought to them rather than sought. That last requirement explains the map. Cold water corals cluster where currents are accelerated and where food is concentrated, which means the edges and canyons of continental slopes, the flanks of banks, the walls of fjords, sills between basins, and the summits and shoulders of seamounts rising from the abyssal plain. Where a current meets a steep obstacle, it speeds up and its layers are disturbed. Internal waves break along the slope, and particles are swept and resuspended past the same patch of bottom again and again. So a coral standing in that flow feeds far better than one on the flat. Their vertical range is wide, from a few tens of meters in the cold water of high-latitude fjords to well beyond 1,000 meters in the open Atlantic. And their lower limit is set partly by chemistry, because the depth at which seawater ceases to be saturated with respect to the mineral aragonite lies much shallower in the North Pacific than in the North Atlantic, and stony corals with aragonite skeletons are scarce below that horizon. The structures they make are not the wave-resistant ramparts of the tropics. A cold water coral reef begins as scattered colonies on a boulder, a dropstone left by an iceberg, an outcrop of harder rock, or the shell of something long dead. And as the colonies grow, their lower branches die and break, and the broken pieces accumulate beneath the living canopy, and fine sediment carried by the current settles into the framework and is bound there. What builds up is a mound of coral rubble and mud, with a thin skin of living coral on its upper current-facing side, and such mounds occur in fields of dozens or hundreds, in some regions rising several tens of meters above the surrounding seabed, and in others buried and rebuilt through successive climatic phases so that the whole pile may be hundreds of meters thick and several 100,000 years old. Individual colonies extend at rates measured in millimeters to a couple of centimeters a year, faster than one might expect in cold water, while the mounds themselves accumulate slowly and intermittently, growing during periods when the currents and the food supply favor them, and standing still or eroding when they do not. Direct observation had to wait for people to go down and look. Crude submersibles from the 1960s onward, working on the slopes of the Western Atlantic, in Norwegian waters, in Mediterranean canyons, and around Pacific seamounts, gave the first views of living thickets, and the descriptions changed the subject from a collection of specimens into a habitat. Observers reported dense stands of branching white coral standing a meter or more above a floor of their own broken skeletons, sea fans and black corals holding their fan-shaped colonies across the current, with the flat face turned into the flow, glass sponges and stalked crinoids among them, and fish resting in the shelter of the branches. Remotely operated vehicles, which arrived in numbers in the 1980s and 90s, could stay down longer, carry better cameras and lights, and be flown slowly along a chosen line, and the video transects they returned allowed cover and colony size to be counted rather than estimated. Towed camera sledges did the same work more cheaply over greater distances, and multi-beam sonar, mapping the seabed in swathes, at last revealed the mound fields as topography, so that a ship could pick out the pimpled texture of a coral mound province on a chart and send a camera down to confirm it. The communities associated with the framework proved to be rich in the way tropical reefs are rich, for the same structural reason, which is that a rigid branching skeleton creates a great volume of sheltered space in an otherwise open place. Surveys of Northeast Atlantic coral grounds have listed many hundreds of associated species, including sponges, hydroids, bryozoans, brittle stars, squat lobsters, crabs, bivalves that bore into the dead limestone, and polychaete worms, one of which lives habitually among the branches of the reef-building coral and adds its own calcified tubes to the framework, strengthening it. Commercially fished species such as redfish, ling, and tusk are found over these grounds, which is one reason trawlers came to work near them, and one reason the corals became known to science partly as broken material tangled in gear. Because their skeletons grow in layers and take up trace elements and isotopes from the water around them. Cold water corals also serve as recorders. Radiocarbon and uranium series dating applied to stony coral fragments has shown living colonies persisting for centuries and dead framework beneath them reaching back thousands of years, while the proteinaceous skeletons of certain black corals and the calcite of some deep octocorals have yielded ages of several thousand years for single colonies, making them among the longest-lived animals known. Cut and polished, their skeletons show concentric growth increments, and the chemistry of those increments carries information about the temperature, the carbon and the nutrient content of the deep water at the time the layer formed, which is a kind of archive that surface sediments cannot easily provide. The corals of the cold and the dark, once fragments in a jar, are now read as structures, as habitats, and as instruments, and the greater part of their distribution remains unvisited, inferred from bathymetry and from the pattern of the currents rather than seen. Reef limestone turns up in places where no sea has stood for a very long time, in desert escarpments, in the walls of quarries, in road cuttings, and in the stone of old buildings, and the recognition of it depends on seeing that the rock is not merely made of shell fragments, but was assembled in place by organisms that grew attached to one another. A fossil reef has an internal architecture. It has a framework of skeletons in growth position, cavities filled by fine sediment and by cement, encrusting organisms binding one surface to the next, and a mass of broken debris shed off its outer slope, and those features can be traced through rock in much the same order that a diver would meet them, swimming from a lagoon out across a living reef crest and down its front. Once geologists learned to read that order, ancient reefs became mappable bodies rather than curious accumulations of fossils. One of the clearest examples stands in the Guadalupe Mountains of western Texas and southern New Mexico, where a Permian reef complex is exposed in cross-section by uplift and erosion, so that the same rock unit can be walked from the flat-bedded, evaporate-bearing deposits of a shallow shelf through a massive reef zone and out onto steeply inclined beds of reef-derived rubble that once sloped down into a deep basin. The organisms that built it were not the corals of a modern tropical reef. Sponges, bryozoans, calcareous algae and encrusting microbial communities did most of the constructional work, with abundant marine cement hardening the mass as it grew, and brachiopods, crinoids and mollusks living among it. Much of the early detailed mapping of the complex was carried out because equivalent buried reef rock in the same region held oil and gas, and the ability to predict where the porous parts of an ancient reef lay had a value beyond curiosity. In the Canning Basin of northwestern Australia, a Devonian reef system has been exhumed by later erosion in a way that preserves something close to its original relief, so that limestone ranges rise abruptly from the plain along what was once the seaward margin of a reef, and rivers have cut gorges straight through the old reef front, leaving vertical walls in which the framework, the cavities, and the four reef slope beds can be examined side by side. Similar exhumed buildups form the peaks of the Dolomites in northern Italy, where Triassic carbonate platforms and their flanking slopes stand as pale towers above darker, softer rock. The mountains and the mineral that makes much of them both carry the name of the 18th century French naturalist who described the rock, and the process that converted the original limestone into magnesium-rich dolomite has obscured many of the finer fossil textures while leaving the large-scale geometry of the platforms remarkably plain. Taken together, the fossil record of reefs extends far deeper in time than the corals do. The oldest structures that behaved like reefs are microbial, built by mats of cyanobacteria and other microbes that trapped sediment and precipitated carbonate, forming domes and columns that grew upward into the water. Such structures occur in rocks of Precambrian age across a span of well over 2 billion years, in some places forming barriers and mounds tens of meters high with flanking debris, and they were, for most of Earth's history, the only kind of reef there was. Animals joined the process comparatively late. The first reefs built substantially by animals appear in the early Cambrian, when small conical, cup-shaped porous-walled creatures called archaeocyaths grew together with calcified microbes into low mounds, and those animals died out before the Cambrian ended, leaving reef construction to microbes again for a long interval. From the Ordovician onward, a different assemblage took over. Tabulate corals with honeycomb-like colonies, solitary and colonial rugose corals with distinctive radial partitions, and stromatoporoids, laminated calcareous sponges that grew as domes and sheets, built framework together with bryozoans and calcareous algae, and their reefs became widespread through the Silurian and reached their greatest development in the Devonian, when carbonate platforms fringed by reefs occupied vast areas of shallow tropical sea. Fossil reefs of this age are known from many regions, including the island of Gotland in the Baltic, the western part of Australia, and the subsurface of Alberta, where Devonian reef buildups form oil reservoirs and were mapped in the middle of the 20th century by drilling through them one well at a time. The late Devonian brought a sharp decline in that reef fauna, and for much of the Carboniferous, the dominant carbonate buildups were mounds of fine mud stiffened by microbes, sponges, and bryozoans, rather than framework reefs of the earlier kind. The Permian complexes, such as the one in the Guadalupe Mountains, represent a later flourishing, and they were terminated by the extinction at the end of the Permian, after which the fossil record shows an interval of several million years with very little reef limestone anywhere. Stony corals of the modern order, the Scleractinians, appear in the Middle Triassic, with aragonite skeletons and six-fold symmetry, and whether they descended from earlier corals or arose separately from soft-bodied anemone-like ancestors has been long debated on the evidence of skeletal microstructure. Their earliest reefs were small, and it took time before coral-built framework again became a common feature of tropical shelves. Jurassic reefs include large sponge-dominated mounds that grew in deeper, cooler water across parts of what is now Europe, alongside coral reefs in shallower settings. During the Cretaceous, thick-shelled bivalves called rudists, some of them shaped like horns or barrels and packed together in dense thickets, built much of the reef-like framework in tropical seas, and they were lost at the end of that period while the corals persisted. Through the Cenozoic, coral reefs recovered and expanded, and the genera that dominate reefs today become recognizable in the fossil record of the past 20 million years or so. With the great concentration of species in the western Pacific and eastern Indian Ocean gradually assuming something like its present shape, the most recent chapters are the easiest to date. Uplifted terraces of Pleistocene reef limestone, staircased along coasts such as Barbados and the Huon Peninsula of New Guinea, have been sampled and dated by uranium series methods to reconstruct the heights and timings of past sea levels. And drowned reef terraces on submerged slopes record the same story from the other direction. The living reefs, meanwhile, became visible in their entirety only from orbit. Shallow tropical water is unusually cooperative in this respect, because blue and green light penetrates several tens of meters in clear water, while red and near-infrared light is absorbed within a meter or two. So a multispectral sensor recording separate wave bands sees bright carbonate sand and pale reef flats standing out from the dark blue of deep water, and deeper structures appear as gradations of tone. The Landsat satellites, the first of which was launched in 1972, provided the first consistent global coverage suitable for the purpose, and a systematic effort to map the world's coral reefs from that imagery produced over years of processing, a set of maps covering reefs in every ocean at a uniform standard, including many small shoals and isolated banks that had never been surveyed from a vessel. Making usable maps from such images requires correcting for the atmosphere, removing the flicker of sunlight glinting off waves, and estimating the depth of water over each pixel so that its brightness can be attributed to the bottom rather than to the column above it, after which the surface can be classified into categories such as sand, rubble, seagrass, algal pavement, and coral. Field surveys by boat and by diver are needed to check those classifications, and their agreement is good for broad bottom types and much weaker for anything finer. Estimates of the total area of shallow tropical coral reef derived from this work fall on the order of 250,000 square kilometers, which is a small fraction of 1% of the surface of the ocean. Newer constellations of small satellites imaging the tropics almost daily at a resolution of a few meters have been combined into a global atlas of shallow reef habitat, while thermal sensors on other satellites track sea surface temperature and are used to identify waters warm enough for long enough that bleaching becomes likely, and radar altimeters measuring the height of the sea surface reveal the seamounts beneath, some of which carry the cold corals of deeper water. We are going to stop here. The music will carry on for a while, and then it will fade on its own. There is nothing more to do tonight. Rest well and see you very soon.